Hey everyone, if you are joining us from the last video where we talked about initiation of DNA replication and formation of the eukaryotic replosome, then in this video we are going to learn about how scientists study processes like these, and where do they get the proteins? How do they study these? And essentially that's what we're going to talk about in this video. So now, one way to study these things, to determine all the different proteins that are needed to replicate DNA in a eukaryotic cell, is to take a tube, a small tube, with a buffered solution here, the buffered water or whatever, and add to it a DNA molecule. In this case, I'm going to add a circular DNA molecule, and that DNA molecule should have an origin of replication. So in this case, I'm going to add an ARS from yeasts. And if you remember from the last video, an ARS is an autonomous replicating sequence. Essentially, it functions as an origin of replication in yeast. And if we put it on this circular piece of DNA and we add the correct proteins to this solution here, well, we should get this molecule of DNA replicated. So we add the ARS to the DNA molecule. Well, we add a DNA molecule that has an ARS on it, and then we add all the different proteins. If you remember some of the proteins from the last video, right, we would need what? The ORC, origin recognition complex proteins 1 through 6. We would need the MCM proteins 2 through 7. We would need CDC6, CDT1. What else was there? There was CDC45, the GINS complex, MCM10. And am I forgetting any others? The DDK, and then there are a whole bunch after these that we didn't even talk about. So after we added all of these, and if we add them in, you have to make sure you add them in the right order to get the initiation complexes assembled correctly. Then hopefully, if we know everything, we'll get this DNA molecule replicated. And then we can do things like, measure the rate at how quickly this is replicated and see if it matches the in vivo or our estimates of in vivo replication. And if it is, well, then maybe we know all the right proteins that you need to replicate DNA. We could also measure the error rate and make sure the error rate matches the in vivo error rate. And if the error rate is a lot higher, then maybe we're missing some proteins that are important for keeping the error rate low, like it is in vivo. So what we're going to do in this video is focus on how do scientists get all of these proteins, because we need to get individual proteins that we think are involved. We need to purify them, and then we need to add them one at a time to see if they are really essential for assembling the initiation complexes and then replicating the DNA. So the technique we're going to talk about is called, well, it's a specific type of affinity purification. Specifically, we are going to focus in on immunoprecipitation, which is a form of affinity purification. So affinity purification, essentially, this type of purification is based on an interaction between molecules. This affinity is talking about interaction between two molecules. And with the immunoprecipitation, with this form of affinity purification, what we would use is an antibody and an antigen. So antibodies are in our immune system, and they recognize antigens. So in this particular example here, antibodies, we always say they recognize antigens, but they're not recognizing the whole antigen. So let's say the whole antigen is a protein. Now, the antibody is going to recognize a very small part of that protein, and that very small part of the protein is going to be called the epitope. So the small part of the antigen that is recognized by the antibody is called the epitope. And so what we're going to do, or what we can do, is we can use an antibody and we can tag a protein that we want to purify with the epitope that corresponds to this antigen here. And then that will allow us to purify the protein. So I'm going to focus in on a very specific example that is very common in molecular biology. So we are going to use an antibody called the anti-flag antibody. Now, flag is an epitope sequence, and I'm not sure if the whole antigen that whoever discovered this flag epitope, I should actually look up the history to that, shouldn't I? But the epitope that is recognized by the anti-flag antibody is called the flag epitope. And the flag epitope is only 8 amino acids. D, aspartic acid, Y, tyrosine, K for lysine, followed by 4 aspartic acids, followed by a lysine. So this sequence of 8 amino acids, this is the flag epitope, and this is what is recognized by the flag antibody. So the flag antibody will bind a protein that contains this sequence of 8 amino acids. You know, as long as that sequence isn't buried in the middle of the protein. As long as it's on the surface or something, the antibody can see it, can interact with it, it will bind it. And again, I'm not sure what is the bigger whole antigen that led to the discovery of the flag epitope, but that doesn't really matter. Molecular biologists, now we just use this sequence right here. We tag a protein with this sequence, and then we use the anti-flag antibody to purify that protein. Okay, so now how are we going to get this onto our protein of interest? So let's see. Let's assume that, let's take one of the proteins from our discussion on initiation of replication. Let's take CDC45. Now, when we're studying initiation of replication, we would need to purify all of these proteins one at a time. And so the way we would do that is we would need to tag this. Tag is sort of a molecular biology jargony term. I'll explain in a moment what that means. But essentially, what it means is we need to put a transgene in the organism, in this case we'll use yeast, that makes a version of CDC45 with the flag tag. The tag in this case is going to be the flag epitope. So let me diagram a transgene that could work for this process. So here's the transgene here. So this is going to be our promoter sequence. And this is where the RNA polymerase would bind and transcribe the transgene. And so this will be the transcript sequences. This is the +1 site. We'll talk more about +1 sites later in the course, but hopefully you remember from genetics, +1 site, this is where the transcript of the RNA polymerase starts transcribing. And we'd have the first ATG, the translation start site, somewhere down here. This is going to be our 5' untranslated sequence in here. So it's part of the transcript, but it's not translated, so it won't be part of the protein. Now down here, we're going to have the CDC45 coding sequence. And let's put a stop code on right here. It could be what, TAG, could be TGA, or TAA. And then this will be the 3' UTR, which will be found at the end of the messenger RNA. And then we'll have some termination sequences over here to help that RNA polymerase II dissociate from the DNA template. Now, in here, this is where we need to put our 8 codons, right? For what? Our 8 codons for the flag epitope. Which was, what was the flag epitope sequence? D, Y, K, D, 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 K. Okay, so there is no stop codon. No stop codon here. So as a result, when we put this transgene into yeast, RNA polymerase II will bind here, it will transcribe this as one long messenger RNA, and then it will be exported to the cytoplasm, and then the ribosomes will bind into somewhere in the 5' UTR, and they will start translating. It'll start translating right at this start codon here. It will translate the flag epitope, and then it will go on, it'll keep going, it'll translate the CDC45 codons. And what we'll end up with after translation is a fusion protein. So at the very beginning here, we'll have flag, and then down here, we'll have the CDC45 protein. Now, some of you might be wondering, will this function? Most of the time it does. Most of the time it does. This flag, it's just in the beginning, and it usually doesn't interfere with the function of the protein. And sometimes scientists have to take a look and come up with various experiments to see if it does interfere with the flag, this little extension of this protein here, to see if it does interfere. But most of the time it doesn't. So this is the fusion protein. What are some other... It seems like I spelled that wrong. Protein. I need spell check. Protein. Oh my God, I can't believe I can't believe this. I knew that didn't look right. This is the fusion, this is a fusion protein between the flag epitope and CDC45. Now, we can also say this is the tagged version of CDC45. We can also say this is the CDC45 that's been tagged with the flag epitope. Essentially, these are joined together. Okay, now this is going to allow us to purify, easily purify the CDC45 protein. Now, how are we going to do that? Well, essentially, what we need to do is take a host cell, like Saccharomyces cerevisiae yeast. We need to insert the transgene, which in this case will express the flag CDC45 protein, the fusion protein. Then we have our host cells with the transgene. What we need to do is grow these host cells, and then we need to isolate all the proteins. And then we end up with a total protein solution. So, well, a solution of all the proteins from the yeast cells. And then we need to perform our immunoprecipitation. And that will give us our purified flag CDC45 protein. Okay, now how do we do the immunoprecipitation? So there are many ways to do this, and the easiest way, which is I think pretty fun, I've done this a couple times, is we use these magnetic beads. So I have that diagrammed over here on the right here. So this is a magnetic bead, and you can buy these from companies, and these magnetic beads are conjugated with commonly used antibodies in molecular biology. In this case, this magnetic bead is microscopic, it's tiny, right? You can't see it without a microscope. It's conjugated to an antibody. In this case, the antibody is the anti-flag antibody. So it recognizes the flag epitope. And then here's our fusion protein right here. So maybe you can't see the words, but essentially our fusion protein looks like this. Flag epitope, CDC45. Well, I have that diagrammed here. The flag is this purple little ball right here. The CDC45 is here, and they're linked together, right? Because they're a fusion protein. So the anti-flag antibody recognizes, binds the flag epitope, and as a result, it's going to it's bound to the fusion protein. It's going to help us purify this whole thing. It's going to help us purify CDC45. So I have that diagram down here. Now, this is our solution of our total protein. We've added the magnetic beads. And what we're going to do is we're going to hold a little magnet down here. All the magnetic beads are going to be stuck at the bottom here, and they're all attached to the CDC45 flag fusion proteins down here. And then we have all the tens of thousands of other proteins. Well, if this is yeast, it's going to be what, 6,000, unless, you know, splice variants too, so maybe more. But let's say 5 to 10,000 different proteins in here, and they're just floating in the solution. They're not interacting with the magnetic beads or the antibodies down here, or the flag antibody down here. And so what we can do now is just remove all this liquid, then add some fresh buffer, and we'll get something like this. Whoa, where's my other? Okay, here it is. So a solution with just CDC45, the flag tagged version of CDC45, and the magnetic beads. Now we might have to change the solution a few times here to get rid of any residual other proteins that were here, but we can wash this a couple times. Wash means change the solution a couple times, so really reduce the number of contaminants in here, so we can get our flag, our purified flag CDC45. Now, how are we going to separate this? Well, we can do that there are different ways to do that, by changing the salt concentration. I think will break the interaction between the antibody and the fusion protein. Also, I think pH might have a role. So just by changing the buffer, you can break this interaction, and then you can just remove the supernatant here, and then your purified protein will come with the supernatant, and the magnetic beads will stay at the bottom. Okay, so that's essentially it. So immunoprecipitation, pretty easy if you can get your transgene into the organism, into the host cell, and get your total proteins. Well, the immunoprecipitation is pretty easy. So the hard part would be, let me see if I can go back to, where's that? Would be doing that for all of these proteins, right? There's 4 in the gens complex. There's 1 here, 1 here, 1 here. 6 of these over here, 1 here, 1 here. And plus lots of other replication proteins that we didn't even talk about. With DNA polymerase delta and epsilon are pretty big proteins with multiple components. So you're going to be doing a lot of immunoprecipitation to make this work. But once you have them all, well, you can add them one at a time to a piece of DNA in a solution and see, as long as that piece of DNA has an origin, has the sequence that functions as an origin. Well, if you have the right combination of proteins and you have all the right proteins, well, then you can get that DNA replicated, and then you can have assemble a good model of how DNA replication works in the cell.